Okay, thank you, Peter. And it's a pleasure to see so many of you here, bright, early morning in London. And we are pleased to present now the case 6, the biotech case. So unfortunately, Preben had some family emergency, so he had to stay back in Denmark. So unfortunately, you will have to bear over and listen to me instead. But fortunately, Steffen will come on the stage every now and then and give the regulators point. And here's the team who has worked over several phone calls and one face-to-face meeting on this case study. And I believe we have all the regulators in the audience. That's a great pleasure. And from Novo Nordisk, unfortunately, several couldn't make it, but we are fortunate to have Torbjørn Strøm Hansen, who is our sort of quality risk management specialist on the panel today. Moving right on into an introduction of this case study, I'll be talking a bit about the product, about how we have approached it, a little bit about the quality risk management, and then we have 5 discussion topics for you, as outlined here. A lot of them dealing with how to implement a model, and this is probably where this may differ from some of the talks yesterday, and also in that we're trying to address what are some of the specific concerns that we may have when we're talking about a biotech drug. So Parenteral alpha is the drug here in question. It is a recombinant human factor 7 analog, and it's engineered to have increased activity. This activity actually is a proteolytic activity, and you'll see later why that's important. It was intended for a bypass therapy for hemophilia patients with inhibitors to either factor 8 or 9. The manufacturing process, you'll see later, is a traditional biotech process, and we included a design space for the activation step. We developed all the other steps as traditional set points with PARs. So unfortunately, late in phase 3, some patients developed anti-drug antibodies, and we had to terminate the project. Because it was terminated so late, we had numerous interactions. I believe we had 2 times interactions with the European system. We have been talking to the FDA, the PMDA, Health Canada, and other authorities. So my introduction is primarily focusing on the European side, but also we can touch upon a few of the others. So in the group, we had discussions around how does biotech differ from small molecules? And although there's a lot of the principles, which are, of course, the same principles, there are some aspects around the complexity of the molecules, which makes it sometimes a little bit more different, and sometimes some caution has to be applied. So often these drugs are glycosylated to various degrees. So we have a mix of species, and in the degradation they can form either active product-related substances, or they can degrade into impurities, and which may be partly active, not active, and some of them may raise safety concerns. So it's one key CQA which we would have liked to put on the list of CQAs would be immunogenicity. That sort of is, I think, an overriding concern in the biotech arena, whether we introduce inadvertently changes into immunogenicity profile. But unfortunately, that CQA is not we cannot measure it directly, and therefore we look into what are the changes that happens to the molecular species, and not always have we all CQAs in place early on. Changes to the manufacturing process may affect product quality, and we believe that a release just against specification is not really a viable option in this case. I think it was speculated yesterday that if you could assay every drug unit, we could release. I think that for biotech it would be particularly difficult to pursue this approach. So of course, an appropriate control strategy has to be in place so that we can ensure the product quality for its safety and efficacy. Here's an outline of the drug substance process applied. A traditional CHO cell culture with the capture chromatographic steps, some treatment, and then the activation step, followed by virus filtration, and then we have the drug substance. So quite typical for biotech. I mentioned to you that this was engineered to have higher activity, which means higher proteolytic activity. And a key constraint in designing the process is the autodegradation of the compound. So activation of the protease and autodegradation is our key concern. So all the steps, the early steps have been designed to minimize the activation and degradation. And then we have the activation step designed particularly then to take the whatever was not activated into the activated form. I'll also use this slide to tell you a little bit more generally about the approach that we are taking at Novo Nordisk for QBD. In all our process and product development, we do apply the principles of quality by design. We're doing lots of DOEs, and I think in the biotech arena, we generally have been doing DOEs even before the Q8 was issued to understand the processes. We have, however, in our company been a little bit reluctant in actually putting these into the submissions. Early on, we said, well, this is going to be difficult. And there was one particular concern. We supply our products to patients in more than 150 countries around the world. So we have a few regulators to deal with, not only the esteemed colleagues in Europe, but around the world. And we were sort of hesitant seeing that complicated QBD would fly in all territories around the world. So we took probably a sort of a cautious approach to this, primarily for this global reason, but also maybe a little because we wanted to see how is this going to pan out and is this going to be difficult. And so our current approach is that we'll include the QBD in the submissions if we see a business case for doing so. So does it make overall sense? Does it give us improved benefits? And you'll see later why we decided to include the QBD elements for this particular activation step. So the purpose of activation step, I think I mentioned that it's really to get it activated, so we have the active drug, but also to minimize the degradation. And the activation is described by a first principle mechanistic model, which makes for some interesting discussions and some opportunities as well. First, a little bit of our quality risk management. I don't think you have seen this slide, but you have seen the principles, of course, from the patient needs and the quality target product profile, or the CQAs and the various process parameters and the control strategy, sort of linking the entire, how is the facility run until what is the product that the patient takes. By way of example here, you'll see that the efficacy is of course important, the dose is important, and therefore bioactivity is important. And in the case, I'll show you how we then have the initial degree of activation coming into the activation step is important, and that we are assessing by an at-line bioanalyzer. This is an example of how we are assigning criticality to the CQAs or the severity class. We are using a 1 to 5 range definitions, and it had been, I should probably have, sorry, and here is an example where we would include some descriptions of the CQAs, see to which extent these are relevant for the drug substance, for the drug product, our severity score, and a bit of rationale why we had this, have assigned the severity score. Now, one of the case studies yesterday had a lot of tables, maybe too many tables, but we would have had this table and I think 2 more tables. One table here which shows for each of the CQAs what are the process unit operations that affect this CQA. So you imagine in this table downwards you have the different CQAs and the drug substance process on the top. And as an example, the activation step that we are talking about would be the amount of the API, it would be high molecular weight proteins, which is affected by this step, and also the degradation, here called the heavy chain degradation forms, is a critical quality attribute affected by this particular step. So our risk assessment is really based on the FMECA methodology described by several of yesterday's presenters. And as also noted yesterday, we see this difference between the criticality, which we can sort of not go away, but we can minimize to a huge extent the associated risk. We have developed a process where we look at the, here says reasonably and realistic failures. So we are not looking at what happens if we have a major earthquake in Denmark, but we are looking at what does go wrong in a plant, in a setting. And we are trying to then assign, does this particular failure mode, this process step, does it impact the CQA? And then we have assigned a number from 0 to 3 on the impact of this particular failure on a CQA. We have the severity score for the CQA, and then we multiply these and get what we have called the SI number. We have had in the group some discussions, and maybe we'll come back to this, about the famous 0 here. Can we have a 0 impact even if we have a very high severity score? And let's get some discussions. As we never, unfortunately, got to submit the drug for approval, we don't know where this would have ended. But maybe Steffen will come back. So following this, we transformed the SI numbers, as you see here, into a criticality value from 0 to 5, and then apply the FMECA. I don't know whether you can see these details from the back or on your screen. Can anybody read the details? Show of hands. Can you see the details? Yeah, okay, it's good. I'll not go through all the details, but at least you can see it. So we're looking here at a failure mode if pH exceeds set point by more than 0.1. The reaction is fairly sensitive to pH, so this is a failure mode that we are interested in. In the next step here, you'll see that we have the relevant CQAs on the top with their severity score. We have assessed impact. You'll see on the 2 left ones, an impact 2 and 3, to whether the pH here will affect these CQAs. We've multiplied. As you can see, we obtained the SI number of 6 and 12, and we carry the worst one, the 12, forward into our further work. So the too high pH could lead to a loss of activity and heavy chain degradation. And the SI of 12 gives a criticality of 4. We looked at what is the likelihood of this occurring. And on a scale from 1 to 5, we assigned a 3 on the occurrence of this failure mode of the higher pH. And we also looked at, is this detectable on the CQA? And because heavy chain and activity would be on the drug substance specification, we give this a rating of 1. This is clearly detectable. What could happen to the CQAs? And we multiply these, we get a 12. Because of our ability to measure this, we said this is in control. Our control strategy is to measure this should anything go wrong, and no further action is needed. maybe by way of adding a bit of a story, you can see we had an occurrence number here of 3. We had underlying a little bit of definition. What did this mean? And we had discussions with an agency that is not the European agency, but a different agency, who wanted really to understand and us to document how did we actually design the occurrence of 3. What was our experience in current biotech manufacturing of pH exceeding set point by more than 0.1? And then we sort of started pulling our hairs, because then, well, how well do we track if the pH set point is exceeded by more than 1, in particular if pH is not that important in the process? So it was getting a little bit difficult if we were to give lengthy, I think, documentation for why each of these scores were assigned. But I think Steffen will address a little bit how you expect that we do this work. So in total for this one step, the activation step, we looked at the 27 failure modes and assessed those. We then plot them into this heat map. On the y-axis, you see the criticality. And on the sort of x-axis, you see the multiple of the occurrence and detection and the way it's done. So high occurrence and low detectability will give a high score. On the other hand, lower occurrence and high detectability will give the low score. Unfortunately, in this case, we're up here. We had criticality of 4, and but we are in the low end. Our approach would be that nothing in the red, in the major risk, would be acceptable. We would not really work further on those in green, and those in yellow we would assess in more detail and say, are appropriate mitigations in place in the control strategy. Moving right on to the activation step, and now it becomes a little bit more close to this model. So the activation is sort of a self-activation and autoproteolysis, where the protein is activated, and but it can also subsequently auto-degrade. The process is quite pH dependent and shouldn't go too low, then it becomes inactive. Now, the input stream is coming from the last chromatographic step, and there will be variability in the concentration. And of course, in such a reaction, concentration is a factor. And also, there could be various degrees of previous activation of the protein. So we're looking at what is the initial degree of activation. And then the time for activation would then depend on these incoming parameters, except we actually found that surprisingly temperature within the acceptable range had limited effect or no effect. Our aim with the model we developed was not to meet the degree of activation at the last decimal, but really to ensure that we met the drug product specification. And this shows how we developed the first principle model. We knew the pH dependency because there is a histidine in the active site, and the proteination of this is important for activity. And also the autodegradation being catalyzed by itself, you can see the concentrations will play a role. And overall, one can derive this model, and I will briefly walk you through it. So the level of activation is here called AKT, and it depends on the initial activation, which is here the AKT0, and then an expression which depends on T time, a constant which is concentration related, a pH dependent factor, this XB, you can see it's shown down here, and then the initial concentration. And then here it comes again. So this is sort of the solution of the mechanistic model. And when we can then solve this, and you can see here this is a function of time, which means we can also solve at which time will we reach a degree of activation of 95%. which has been our target in the sort of in the center of the drug product or drug substance specification. The pKa influence is described here, where we at 7.6 have the, I guess this would illustrate the half protonation of the histidine group. So, having developed the model, our first approach was then to calibrate the constants. We needed to set 2 constants in this model, and that was done in a lab scale, and here illustrated at a fixed pH, but at a different concentration, and you'll see how the data points and the curves seem to have a reasonable fit. This is for the nerdy biochemist. It is a 406 amino acid protein with, I believe, 12 glycosylation sites. But of interest here is that it gets activated by clipping itself in this position, but it can further auto-degrade by further clips in these positions. This graph also illustrates with the blue arrow actually the 3 amino acids which we changed from the native human factor 7, which gave rise then to antibody development in the patients. The native, the factor 7 that we currently produce and have on the market, we do not see these antibody formations at all. This graph illustrates where if we prolong the activation too long, we get these heavy chain degradation elements on the left-hand side where we follow the time. On the right-hand side, we have plotted the heavy chain degradation versus degree of activation. And you can see once we hit the very high degrees of activation, we start to get the heavy chain degradation. And of course, our aim is here to end at a place where we have significant activation, because the active protein is our active drug, but we also want to minimize the heavy chain degradation. And here's our design space, and it's because being a model is a bit unusual. So I'll try to walk you through it, and then, of course, you can feel free to ask questions. So we have the model on the right-hand side. Now, this time it has been solved for time. So this is the time it will take to achieve the degree of activation desired at a given pH. We have down here, we have what we call the boundary condition. So this is where the model is valid and where we will operate it. So we will look at degree of activation from 10 to 99. We're looking at reasonably narrow concentration rates from 1.4 to 2.1 gram per liter, and a pH from 6.2 to 6.8. Reaction time is actually what we would vary, which would be the output of the model. And within this temperature range, as I said, we see little temperature effect. The graph here shows the concentration and the initial degree of activation. And then there are surfaces at 3 pHs for what the time needed to achieve the activation. So we could plot in here the measured by the bioanalyzer initial activation, the desired final activation, and we would use 95%. This would give this equation, and out would come the time needed to run the activation process. Sort of feed-forward use of the incoming conditions using the model to calculate reaction time. And as you can see, there's quite some pH dependency here. And obviously, if it's very much degraded already coming in, we need less time than if we have a low degree of initial activation. So we had the model, we calibrated it, and then we had the discussion, how should we verify this model to confirm that it works? And we'll also, in this point, discuss what is the scalability. So for the calibration and verification, the blue dots here, if you look at the cube to the upper left, this shows the incoming parameters, pH, the initial activation and the concentration. And in this three-dimensional cube, you can see how we did on the blue ones, the calibration runs, and then we had some clinical material manufacturing that we used for the verification. The three other graphs here actually shows a view from each of the three sides of the cube. So you can see that on the pH and activation, we used pH from the low to the high end in our calibration. We had primarily low activation in our calibration part. And looking at it also where we can see the concentration, you can see that we had concentration points spanning this area. Also see that when we then ran the clinical manufacturing, we did most of it at the center pH. So we showed this to regulators and we said, we can just pick one point and we can see whether it works, and then we have verified it. Well, life is not that easy. So we got a bit of feedback. Well, we think you should verify this a little bit more. Big surprise. And we felt it was easier to do it than to fight it. So we went back and did some primarily lab scale work. And this is sort of a reduced factorial design that we have used. And also so run these experiments shown here. And you'll note also that we had discussion, is this really true about the temperature? So we have, if I can find it, we have temperature which are also outside of the range, slightly outside of the range, to see whether this would also affect it. So Steffen, we did these, and the model was verified. I think we got very good results confirming here the sampling versus the predicted lines. So the next question was then, what about taking this from the lab to commercial scale? What would be needed? And well, this is all in solution, taking place, reasonable temperature. The only thing which might potentially, we felt, be scalable would be the pH time for pH adjustments. But as this is fairly short compared to the reaction time, we said, well, this we really don't believe is a factor. So in practice, this is scalable. And therefore, we would test it in the normal operational set point and see if the model work in the large scale. And I think Steffen will comment on whether you agree to this. And I'll show you the data that we then actually obtained. So here on the x-axis, you will see our scale-up factor. So the lab work is done in the milliliters, and the commercial scale is like the many liters scale. And on the y-axis, the center axis here at the 0 point here is our aimed final activation. And as I told you, this would normally be 95%. And also told you we have a spec, or maybe I didn't, but the spec is from 90 to 99. So on this scale, the spec would be from the lower -5 to +4%. And as you can see, all our experiments here, they meet that condition. And maybe there's a tendency that once we move up into scale, maybe there's a little bit more robust. But obviously, we have few numbers, but we conclude that this is scalable, and we do not need to further show scalability. Now, over to you, Steffen.
Yep, thank you. Thanks a lot for this really nice introduction, and I would like to give you some regulatory feedback from just specific for this case, and some, let's say, more general regulatory view on some of the points. And some of the points were identified yesterday already, but we really try to focus on certain aspects with regard to quality by design. And as you already mentioned, we were really happy to see these tables. And talking about tables, this is exactly what we would expect, that you have characterized your product, you have identified the critical quality attributes, and then you characterize which manufacturing process might impact which quality attribute. And this is a term which was not used yesterday. These are so-called worst-case linkage studies. In your case, it was quite simple. You have one manufacturing step. You try to apply a quality by design. It's just the activation step. You have two defined parameters: pH and temperature and reaction time. And this is quite simple. You can do this. You have performed these small-scale studies, but it's getting more complicated if you have more manufacturing steps, if you have more, let's say, process parameters you have to look at, and then you have really to investigate which process parameters do influence which critical quality attributes. And you have mentioned all these different, let's say, post-translation modifications. You might have this glycosylation, you might have, let's say, other impurities, you might have oxidation, and you have to address these things. And I think the tables you have provided in the beginning of the presentation were really helpful to understand that you have really looked into these things, and we would like to see these tables. But actually, the modeling, and this would be the question I would ask you, whether you have performed these kind of experiments here indicated in the table below. This is just a copy of your table. We would expect exactly these kind of studies, that you perform small-scale studies. I guess it was my expectation that you will not perform all these different conditions under large scale, but we have learned yesterday that even that is possible, that you perform at least 30 runs at large scale to let's say verify a certain model, but we would expect at least small scale studies to address the worst case conditions. You should be able to characterize your product. You should be able to expect what happens if you change the parameters, and if you have defined this, you should be able to let's say, expect what happens to your product. And then we would expect that you have a kind of modeling or an experimental design that you really take all the worst case conditions into consideration and use at least an experiment where you have set all the parameters at the worst case conditions, and you see whether your product is still within the expected range of the quality attributes. And this is exactly what you have done with this table down here at the bottom. And even you have conditions where you did not meet your expectations, so you did not meet your critical quality attributes. And that is, I guess, the best thing you could do, and we were very happy to see these tables. But this is a general remark I would like to make. These are exactly the data we would like to see. And Jean-Louis yesterday just remarked that it's not only data review, it's more scientific knowledge what we should look at. And I agree with you, we should have a scientific discussion, but we actually look really at the data. So we will still do some data review. We will look at these data, at the results of the, let's say, small scale studies, and this is exactly what we would like to see. Whether we would like to see all the design of experiments, I'm not sure. You have just mentioned you have performed many design of experiments. I guess it will be too much to review. So we will end up with thousands of pages, and we will not look at all these data. But at least we would like to have addressed, let's say, the worst case conditions. And as I said before, in your case, it's quite a simple example. You have just these two parameters. You have just one critical quality attribute. But if you're talking about a more complex molecule, you look at all the different variants which could appear. This might really get complicated. Another topic is the verification or scalability of the model. We discussed this quite intensively, and we said it will not be possible if you have a multivariant, let's say, investigation to verify it at commercial scale. But as said before, it might be possible to perform it at a commercial scale as well, but we would not expect this. So you have to convince us that your model is scalable, and the model verification at commercial scale is done actually under normal operating ranges. and not under worst case conditions. This is one of the topics one could discuss, whereas the scalability of the model should also take place under worst case conditions, but our current understanding is that model verification under normal operating ranges is acceptable, and you should follow on during the post-approval life management, also the scalability of the model. So the approach you have taken was acceptable for us, and then I would like to hand over to you again.
Okay, thank you, Steffen. I'm glad that you didn't get any good ideas from one of the cases yesterday. It was a legacy product with, I don't know, 30 batches in commercial scale. Don't expect that for a new drug, please. I'll briefly actually go, I thought I had sorry for this, I'm going the wrong way. So here we have the model again, and as you can see, we have a boundary condition on pH from 6.2 and 6.8, and I think we have shown that we have also tested different pHs. So we're looking at pH, and we believe that we can change the pH within this set boundary conditions without regulatory consequences. We also are looking into, in practice, maybe we could use this model a bit differently. So we had solved the model so that we calculate reaction time as a function of the input parameters. But we could also do it a little bit differently. Maybe in the daily operation in the plant, it would be better to always have a fixed reaction time and then just adjust the pH so that the reaction time was always this fixed one to achieve the to get the expected activation degree. So therefore, we are looking here into, to your left, you see how we have presented the model so far. To your right, it's this alternative approach in daily life, which would be, say we will fix reaction time and then just adjust the pH so that it fits. And this, of course, we would very much like to be able to do if we end up in practice, this being a better solution.
And this was one of the excellent examples how you could use quality by design and how you could use modeling. And in that case, I guess it might be possible to rearrange the model. This is just the modification of the control of the activation step from a fixed pH and a variable reaction time to a fixed reaction time and a variable pH. And this would give you more, let's say, more insurance for your manufacturing process. You can blend better maybe, and as I said before, it would be acceptable for that specific case there's a clear interdependence between pH and reaction time. We have a valid model, and therefore this rearrangement seems just to be a mathematical calculation. So I guess for this specific case it might be possible, but never as a rearrangement of models for more complex, let's say, manufacturing steps is possible, is always a case-by-case decision. But how to deal with these rearrangement of models, this might be an issue for the discussion later on, whether this rearrangement should be, let's say, reported to the regulatory agency, or whether even this rearranged model needs a verification at small scale, at large scale, or can it only be a continued verification, so design-based verification. So these are open questions which we should address maybe during this discussion later. And then we have the next discussion topic, and I hand over again to Henrik.
So this discussion topic is related to, we have this bioanalyzer at line. We are measuring the incoming degree of activation to our process step, and we're measuring the outcome when we have conducted the reaction. We are confirming this also with the bioanalyzer. This in reality means that for each run we will get one new set of information, and could we over time improve the precision of the model by feeding in the new information into calibration of the constants. And therefore, would we be allowed to recalibrate the model based on the more information that we will gain by doing more and more runs? Otherwise, the model was the constants were calibrated based on reasonably few lab scale runs, but now should we use the increased knowledge and also in full scale to actually on an ongoing or an intermittent basis to recalibrate? And how should this be done? Is this that if we see a systematic deviation that we should go in? And this first approach would be to do sort of a simple adjustment. But I think the heart of the matter is to which extent would we be allowed to perform a recalibration of these constants under GMP. And that's, of course, our approach, that this is only a matter of GMP, and you can inspect that at certain occasions, but it's not something that we would have to submit for variations. Well, what do you think, Steffen?
I guess here we have to make more general statements. Regarding your question, it was a notification, or it was a better, let's say, a better fitting of the model. And if you have a changed model and all the data you have gained and provided, and these results in a better or unchanged fit of the actual data and a better model, I guess there is no need to notify the authorities. This is the remark with respect to this case. But now I would like to make some remarks about the notification of regular authorities, which kind of, let's say, changes should be reported to the authorities. And the first bullet point there, movement within the design space, we have talked about yesterday about this case as well. As long as you're moving within an approved design space, there is no, let's say, notification of the regulatory bodies required. But if you have to adapt the models, you have to review the design space, you have to, let's say, review the model validity, the criticality of the quality attributes and the process parameters, and how often do you do that? And this is a very important question, and I guess you will not find this information within the dossier. It will be impossible. But the review period is really an important issue. So it should be short enough that you could react quite fast, but it shouldn't be so long that you, let's say, will not notify any authority. And this is the next slide. How should we deal with that? I mean, GMP inspection, and this was mentioned yesterday as well, are a really good option to look into, let's say, these systems. You have to monitor your review, you have to collect the data, and you will not provide all the data to the agencies. But we might have the possibilities to look at these data during GMP inspections, and actually this was really stressed yesterday. We really encourage assessors to go into a GMP inspection with the inspectors and to look at these systems. And the quality by design approach will require these interactions between assessors and inspectors. And we have made the proposal that the product quality review might be a really nice tool to notify the agencies, regulatory bodies about certain changes. Because with this product quality review, you have all the information what you need. You have change controls, you have deviations, you can, let's say, review, or you can report your review of the quality by design or the design space. So we think the product quality review would be an ideal, let's say, tool to, let's say, report certain changes, adaptations of models, adaptations of quality attributes, adaptations of process parameters to the agencies. And I guess this is one of the points which can be taken also by EMA, where such a product quality review might be a regular document which could be provided to the assessors. This is just one of the examples how certain process parameters could be reported to the agencies. We are talking about non-critical process parameters, and then you have a risk assessment, whereas a non-critical process parameter remains non-critical. If the outcome is acceptable, then there's no need to inform health authorities, but as soon as you change a non-critical process parameters to a critical process parameter, how should the agency notify it? And this is really important. And This is taken out from the variation regulation. Also, variation regulation was mentioned yesterday. If you're moving outside of the design space, you have to submit the variation anyway. It's a type 2 variation, and then you have informed the authority about the changed, let's say, conditions. But what about non-critical parameters? And we just realized there was an option within the variation regulation at a certain time that if you change a non-critical process parameters, you could submit a type 1a variation. This was quite a nice approach, but unfortunately this option was taken out. And we should really think about how to notify authorities about these kind of actions which are taken by companies. Because sometimes who is responsible for the evaluation, what is considered non-critical. So personally spoken, I would prefer if I would know what happens within the manufacturing process, and I would prefer if we would have the chance to have this, let's say, this type of variation again back in the variation regulation. And then I guess I have to proceed. I would like to present some other viewpoints and additional discussion points, what we have seen during the evaluation of quality by design applications for biotech products. And the first topic I would like to address is the criticality of quality attributes and process parameters from our point of view. So if you're talking about quality attribute criticality, how can we be convinced that you have addressed the right issues? And I guess also here we can just talk about an inspection to see on-site who took part in the evaluation. It should be, let's say, an expert team who's reviewing the molecule. So this review team should include people from research development, should include people from manufacturing, should include people from QA. So it should be a team review, and this we will not see in the submission, but this you can see during inspection. One important factor is the impact scale. How is the impact scale developed? And you have shown this nice table. That's why I said we prefer to have these tables, but these tables should be really explained. How do you define a low critical impact or a high critical impact? This is really important to follow on. And talking about harmonization, and it would be a nice idea to have a kind of common impact scale across companies, but also companies now develop their own impact scale. It's their own manufacturing process, of course, but maybe we can develop in this direction. We can have developed ideas to have a common impact scale that we could expect what is meant with this impact scale. As said before, we need to have a cross-functional committee to have input from all the, let's say, different sections for research development, manufacturing, QA. And then we have, of course, touched this scale issue already, the risk score. Can it be 0? And I guess this is a topic which we might discuss later on, whether there might be a risk factor which can be set on 0, even if you have a high impact, for instance, but your uncertainty is really, really also high. Can it be then that you have a risk score of 0? Is this possible? But we can discuss this during the discussion later on. Identification of critical process parameters. As before, I mean, you have to characterize your product. You should know what happens if you, let's say, screw on your manufacturing process, what happens to the critical quality attributes, and you need small-scale studies. And I guess we are satisfied with small-scale studies as long as you can convince us that these studies are scalable. And I think a really worthwhile approach is to use worst-case linkage studies. This is an ideal, let's say, situation which you could provide within the dossier. And if we have these kind of studies, these kind of results, I guess we will be satisfied. Parameter definition, this is one of the hot topics, I have to say. What is the critical process parameter? Actually, a critical process parameter is any parameter which has a practical significant impact on the critical quality attributes. And we have always the discussion with industry, with companies, whether an adjustable parameter of the process that can be maintained within a narrow range is still considered critical or non-critical. We think, from our point of view, from the regulatory point of view, that as long as this parameter has an impact on the quality attributes, it should be considered critical. I know that the view of the industry is different, but this might also be a discussion point later on. Control strategy. Control strategy was touched yesterday quite intensively. For biotech products, I guess there must be a minimum control system in place, not only to ensure the quality of the product, but also to monitor the consistency of the manufacturing process. And I guess consistency is also one of the, let's say, issues what we might discuss later on during the panel discussion. We not only want to ensure that the quality of the product is given, we also want to ensure that there is a consistent manufacturing process, because the process still defines the product quality at the end. And how to do that? The control strategy comprises all the different elements, including the control of raw material. So raw material control, I guess, wasn't taken as one of the points within the quality by design submissions we have seen so far. So we have seen quality by design for certain manufacturing steps, but we haven't seen any approach that also the raw material might be let's say, a point for quality by design. You might use different sources, different let's say suppliers. So this could also be a part of quality by design. You should have process controls in place. You have to define in-process controls. You have to define let's say assays, specification for lot release, and also for stability testing. And then it's quite important which kind of tests are done for release and which kind of tests are just done for monitoring, and which kind of tests might be reintroduced in a certain manner for comparability, let's say, purposes. So, and this brings me to the next topic, real-time release testing. Could it be part of the control strategy for a biotech, let's say, product? Currently, I think it's really difficult to release a biotech product only based on process, let's say, controls. But we are moving in a different direction. And when I have said this, I guess 5 years ago, for instance, for host cell protein, host cell DNA, host cell protein was always part of the release specification for drug product. But then we moved these specifications, for instance, towards drug substance. And meanwhile, it is well accepted that these kind of specifications, or not specifications, but these kind of assays are performed during the manufacturing process. So host cell protein, host cell DNA are now generally accepted as, let's say, in-process controls. They are not expected, at least not for all the products, for drug substance or drug product. So we are moving in this direction to have a reduced testing. But I have really doubts that we might end up releasing, let's say, a biotech product without a final release testing. And therefore, with these words, I would like to end our presentation. I guess we still have some time for specific questions. For this case, I guess we have time for, we would like to ask you to ask specific questions for this case. And these discussion points listed here should be taken into consideration later during the panel discussion. So the floor is open for questions.
Yeah, we have a question over here.
Hello, I'm Emil Swan with the Swedish Inspectorate. I'm very interested in the idea of recalibrating the model. You would obviously have new ground included in the approved design space or approved volume you can exist in. But you would also, when you recalibrate, exclude previously approved volumes. How would you, within the quality management system, propose to act if you have a batch that was approved on that now excluded volume? That would be
but we would probably not have excluded, but the idea would be that we would take all the data available. So we would not exclude previous data, but we would add on new data.
Yep.
That was the approach. But you could say eventually, if we have done 1,000 batches at the center point, then of course those will dominate the previous few values outside the center point.
But it could, if I don't misunderstand, it could mean that you have batches released according to, in effect, a specification which is no longer valid, and there is no then regulatory interaction around this fact. So would that mean a recall?
I don't think so. When we had these different levels, as I said, we do measure the degradation and the activation. So we will not release batches. but we would optimize the model. And this is, I try to say, when we move into commercial scale, if we have many batches and we take all the data into consideration, of course the many data will weigh more heavy as we move on into commercial scale. So that's the risk, that do we lose the impact of the early data. But still, in practice, it might enable us to hit the target more precisely by optimizing the parameters.
This was a remark I had. This is a better model. If you, let's say, improve your model, then there's no need to notify the authority. But if you are moving out or you have kind of new parameters, you should notify the agency, and then it's a kind of approved. So we will know it, and we will approve it with a new parameter, of course. So there's no batch around which is, hopefully, which is not approved.
No, absolutely not. Graham?
Yeah, Graham Cook from Pfizer. Emil, I think your point that you were trying to make was, if you've changed the model, does that mean that the space that they're manufacturing in is different, and hence you get product which is not necessarily of the same quality? That's I think the question that you were raising, and I think the answer is that what Novo Nordisk were proposing, and Henrik, correct me, was that the specification, the quality of the drug substance doesn't change in the approach. In one approach, as I think you indicated, you are keeping the pH constant and varying the reaction time in order to produce material which was between the 90 and 99% activation. So it's like going from Canary Wharf to Waterloo Station on the Jubilee line, and you get to Waterloo Station. The other approach is to say, okay, for, I guess, manufacturing convenience reasons, if you talk to production guys, they like to plan their production schedules. So to have variable manufacturing times is really, really awkward. It's much nicer to have a fixed process time and predictability. It helps plan production schedules. So what Novo Nordisk appear to have done, which is really quite clever, is to say, okay, we've got this model. We can now, instead of running the process at a fixed pH, we can run it at a fixed time and vary the pH, again to achieve the same endpoint, activation between 90 and 99%. The quality is the same, but instead of going on the Jubilee line from Canary Wharf to get to Waterloo Station, I've gone on the Jubilee line, I've switched to the Northern line, and eventually I get to Waterloo Station by the same route, but I still get to the same endpoint. And that's, I think, what's clever, and that's really illustrates quite nicely the value of the process understanding and the use of a model in order to enable you to make those changes, and it's based on science. So you can be very confident, I think, that you get to the same quality endpoint.
Well, I think I'll leave the London Underground to Graham. And there are two elements. And one is what we mentioned here about, can we change the way we use the model? And the other one was, can we sort of, with more data, are we sure that this number is actually 7.61, or are we having a better fit if it's 7.5? And the other constant here, can we optimize the model by changing that? But we did not intend to move outside the boundary conditions. No way. Or at least if we would apply for a variation. Yeah, more questions?
Sean Jones, MHRA. Regarding updating the model, do you need to have a criterion for a particular batch lot of data? Is it suitable for updating the model or not? So did you, in this particular case then, or have you thought of what the criterion should be for that batch data to be suitable for updating the model? Because the batch data may not be suitable for updating the model. It might be an atypical batch, and you wouldn't want to update the model with an atypical batch. You don't. So how do you know that the batch that you're going to use to update the model is within the normal scope and process capability?
But I would say now, as mentioned, unfortunately, we never got this through the approval process, but we have had some internal initial thinking about what could we define criteria that should be met for us to to refine these parameters. And there we had some early thinking about, could we have a sort of a suitability index that would need to improve for us to actually recalibrate the constants. We have not had a lot of discussion about should we exclude certain batches because them being atypical, or how could we evaluate that? But I think it's a reasonable, fairly point that we should be addressing. Yeah, Frank?
Frank Montgomery, AstraZeneca. It's a question to Steffen. Picked up your point about the variations and the unfortunate removal of the type 1A. And that's very similar to one of the examples that I described in my presentation yesterday. Right. And had a conversation with Christine Moore, and she told me off for not describing the same scenario. For describing the same scenario in the US, and I think the comments that Christine made were really helpful and insightful, because we went into that conversation with FDA. We ended up having a teleconference with them to clarify the issue, and their perspective was that all right, we submitted this as a CBE 30 in the US eventually, which mirrors the type 1B. But after the conversation and explaining the situation, their written feedback back to us was that they would consider this as an annual reportable item in the future. And through sort of quite pained discussion within the agency, considered it analogous to tightening of a specification. And I thought that was quite helpful. But I think it's a key point that we maybe need to take into further discussions later on today.
Actually, this topic came up during the inspection when the company really asked how should I deal with that, and we proposed that there might be options, and I think this product quality review, any product review, is an ideal tool to see these data. And yeah, we proposed it to EMA as well, and we might discuss it internally. We have to discuss it internally.
Yeah, and I suppose one of the aspects that we could consider if we were to adopt this kind of approach is immediate notification rather than notification at the end of the year, because then that avoids significant differences of opinion sort of festering for a very long time.
I suppose one of the main messages I would like to make from the yesterday, or outcomes from the yesterday discussion, there should be an open discussion between the companies and the agencies. There should be some kind of this is a matter of trust. And it's better to notify a bit more than less. So it's we are quite open for scientific discussions, and I guess we will not, let's say, try to. No, I guess. It's not necessarily, the interaction is really important for us, and it solves many problems which might come up.
Just to continue on the last point, I do not know, because I obviously wasn't there, why this item was deleted from the type 1A variations list. However, there's a certain regulatory logic to that. Namely, it's about non-CPPs. If it's a non-CPP, then it does not affect product quality. If it does not affect product quality, it does not belong in the dossier. And if it does not belong in the dossier, then it cannot be changed. So variations also are simply non-existent. I mean, I do not know if this was the thinking, but I see Mats, but I think it relates to a more central point, namely dossier content and what kind of information should be in the dossier.
We would definitely support your interpretation, but maybe Matt has a different view.
No, actually, I haven't. But Mats Wallin, Medical Products Agency. No, I think the thing is maybe you're a bit on thin ice, than Martini, in the sense that. But I think we have discussed it earlier that we are saying we should have a reasonable level of detail in the description, but we are also saying all non-CPPs should be in. Which for biotech, I think you can expand on that Henrik, that we can have 500 non-CPPs. Should all of these be in? They have never been in before. Why should they be now? So this is an issue. So I have sympathy for you. If we can agree, between the regulators and the companies that this is in fact a non-critical parameter, then I would also say that maybe we should be able to handle this outside of the variations regulations, but still being able to see it in annual reports, in inspections, and so on and so forth. But I think this is an issue for further discussion on do we need all non-CPPs in the description?
Well, maybe just a brief comment. I think it's fair if we are challenged at the in the dossier about what is a critical process parameter, we should justify why certain are not. But then at least I think my opinion, the Novo Nordisk opinion is beyond that, then we should not have to report on non-critical process parameters.
But for that, we should have same understanding of non-critical and critical process parameters. But another question over here.
Yeah, Gerd Thurau, Roche and Entech. I wanted to step away from the CPPs for a second, or non-CPPs. Go back to the question about the model and the model update. So I don't know this particular model, but I know that Dor isn't here, so I want to do my best Dor impression and say that obviously it's in the best interest and really best practice among people who do modeling that if there are updates to the model, the data that are included in such an update are, scrutinized for, for suitability. So in a case like this, I would expect that at minimum, the people who work on the model would verify that whatever the model is to predict is actually tested in those batches and they're suitable. So I think that's just best practices. There's an interesting aspect too, if you update a model, whether older batches with the new model would still predict the same and how you deal with that. I think that's something, and that's solved in maybe different kind of models like spectroscopic models. But there's probably more discussion room for that. Since I have the microphone just for another second, I also want to actually comment on what I found is a very, very refreshing perspective from Steffen on the discussion we had yesterday on full-scale verification of models. So I thought this was an excellent example, and I think also, the colleague from Novo Nordisk pointed out, for a new product, it's maybe a different discussion. And also in reflection on some of yesterday's studies, I mean, I don't know the details of these studies from yesterday, which have now been quoted with the 30 batches and the 50 batches. I think it would be interesting to discuss just like the regulatory agencies have a pilot, most pharmaceutical companies also have a QBD pilot. So the initial QBD project usually will have an element of experimentation, an element of doing a lot. And I would caution that maybe what has been presented is sometimes the outcome of this pilot, but not necessarily best practice. So I think we talk about, sometimes, yes, we talk about industry standards. So again, it would be good to discuss maybe in the breaks whether what has been presented yesterday is really what these companies would do today or what other companies would do. So I just wanted to bring this out. I don't know the details, but I just don't know how to let this stand as this is what everybody is doing and should be doing. So that's why I found this example today also very refreshing. Thank you.
Hi, everyone. So this is Manish Gupta from GlaxoSmithKline. So I'll kind of share a perspective to add to what Frank mentioned and the feedback that Christine provided from the FDA. So in our recent experience, we were approached by Health Canada across a few projects where they asked us, so why aren't you actually calling these multivariate ranges based on interactions a design space? And is one of the impediments to calling it a design space the inclusion of non-CPPs and the filing or the post-approval challenges that may come up? So they advised us, well, include the non-CPPs as part of your design space. That's the right thing to do. But then also along with that filing, provide that post-approval change management aspect. So that I just wanted to share that was quite proactive of the agency, and I think the right thing to do.